Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be independent to you. In the last session, we ended up with causes of ketosis. In today's session, we will continue with explanation of ketogenesis, in which we will see the effect of glucagon and insulin. Then we have salient features of ketosis, which includes all the symptoms. Then we have diagnosis of ketosis, which includes Rothera's test and Gerard's test. Lastly, we begin with a new chapter, cholesterol and lipoproteins. Good morning, everyone. Let us start the topic. Explanation of for ketogenesis. During starvation and diabetes mellitus, the blood level of glucagon is increased. Glucagon decreases glycolysis, increases gluconeogenesis, increases lipolysis, decreases methanyl-CoA, and increases ketogenesis. Increased glucagon to insulin ratio is potentially ketogenic. On the other hand, insulin has the exact opposite effect. It increases glycolysis, decreases gluconeogenesis, decreases lipolysis, increases methanyl-CoA level, and decreases ketogenesis. The ketone body formation is located at following three levels. Level 1 is lipolysis. Free fatty acids are the precursors of ketone bodies. So factors regulating the mobilization of fatty acid from the adipose tissue will also control the ketogenesis. Insulin decreases lipolysis, whereas glucagon increases lipolysis. In this diagram, we can see the three levels of ketogenesis. Level 1 includes lipolysis activated and increased non-esterified fatty acid in plasma. Level 2 is increased carnitine amyl transferase 1 and decreased methanyl-CoA, which further increases beta oxidation. Level 3 is increased gluconeogenesis and decreased oxaloacetate. All the three induced increased generation of acetyl-CoA and decreased utilization of acetyl-CoA. This leads to ketone body synthesis. Level 2 is entry of fatty acid to mitochondria. The mobilized fatty acid then enters the mitochondria for beta oxidation. Carnitine acyl transferase 1 regulates this entry. Methanyl-CoA is major regulator of CAT1 activity. In diabetes and starvation, glucagon is increased, which decreases methanyl-CoA, and so beta oxidation is increased. Level 3 includes oxidation of acetyl-CoA. When the above two steps are increased, more acetyl-CoA is produced. Normally, acetyl-CoA is completely oxidized in the citric acid cycle. In diabetes and starvation, glucagon ratio is increased and hence key gluconeogenic enzymes are also increased. When oxaloacetate is diverted for gluconeogenesis, citric acid cycle cannot function optimally. On the one hand, acetyl-CoA is increased. On the other hand, its utilization is decreased. This excess utilization that is of acetyl-CoA is channeled into ketogenic pathway. In both diabetes mellitus and starvation, the oxaloacetate is channeled to gluconeogenesis, so the availability of glucose is reduced. Hence, acetyl-CoA cannot be fully oxidized in the TCA cycle. Coming to the salient features of ketosis. Metabolic acidosis. Acetoacetate and beta-hydroxybutyrate are acids. When they accumulate, metabolic acidosis results. Reduced buffers. The plasma bicarbonate is used up for buffering of these acids. In this diagram, we can see symptoms of ketosis. These are improved cognitive performance, appetite suppression, fatigue, muscle cramps, digestive issues, weight loss, decreased energy for exercise, keto breath, and insomnia. Third is Kusumal respiration. Patients will have typical acidotic breathing due to compensatory hyperventilation. Then there is smell of acetone in patient's breath. There is osmotic diuresis induced by ketonuria, mainly to dehydration. Then there is sodium loss. The ketone bodies are excreted in the urine as their sodium salt, hence leading to loss of cations from the body. Dehydration. The sodium loss further increases the dehydration. Coma. Dehydration and acidosis together contribute to the lethal effect of ketosis. Coming to the diagnosis of ketosis. The presence of ketosis can be established by the detection of ketone bodies in the urine by Rotheras test. Supportive evidence may be derived from estimation of serum electrolytes, acid-base parameters, glucose and urea estimation. Rotheras test. Saturate 5 ml of urine with solid ammonium sulfate, then add 3 drops of freshly prepared sodium nitrochloride followed by 2 ml of liquor ammonia along the sides of the test tube. Development of a purple ring at the junction of the two liquids indicates the presence of acetone or acetoacetic acid in urine. Here we can see the Rotheras test. On the left hand side, there is negative ketone bodies, so there is no purple colored ring. On the other hand, there are positive ketone bodies, so the purple colored ring is seen. Gerard's test or acetoacetic acid test. To 5 ml of urine, add dilute ferric chloride solution drop by drop till a maximum precipitate of ferric phosphate is obtained. This is to eliminate the phosphate which may obscure the color in tests. To the filter, add excess ferric chloride. A red color indicates the presence of acetoacetic acid. In this diagram, we can see Gerard's test. To the urine, add 10% ferric chloride. A Bordeaux red color develops. If the color persists, there is absence of acetoacetic acid and presence of salicylates. On the other hand, if the color disappears or heating, the presence of acetoacetic acid is present. Coming to differential diagnosis of ketosis. The urine of a patient with diabetic ketoacidosis will give positive Bennett's test as well as positive Rotheras test. On the other hand, if patient is starvation, then its ketosis includes Bennett's test is negative, but Rotheras test will be positive. Coming to management of ketoacidosis. Treatment is given to insulin and glucose. When the glucose and insulin are given simultaneously, potassium is trapped within the cells and fatal hypokalemia can occur. Hence, the clinician should always monitor the electrolytes. Administration of bicarbonate and maintenance of electrolyte and fluid balance are very important aspects. Coming to the new chapter, cholesterol and lipoproteins. The word cholesterol is derived from Greek words. Chole means bile, steros means solid, and OL means alcohol. Almost all nucleated cells can synthesize cholesterol. It is widely distributed in the body. In a 70 kg man, a total of about 140 gram of cholesterol is available, which is roughly distributed as 30 gram in brain and nerves, 30 gram in muscles, 30 gram in adipose tissue, 20 gram in skin, 10 gram in blood, 10 gram in liver and spleen, and 5 gram in bone. Cholesterol is a light yellow crystalline solid, but when the crystals are examined under the microscope, they show a notched appearance. Cholesterol is soluble in chloroform and other solvents, fat solvents. It is the most important animal steroid which other steroid compounds are formed. Cholesterol is widely distributed in animal tissues. In this diagram, we can see the cholesterol. Coming to clinical significance of cholesterol. The level of cholesterol is in the blood is related to development of atherosclerosis and myocardial infarction. Abnormality of cholesterol metabolism may lead to cardiovascular accidents and heart attacks. Coming to structure of cholesterol. All steroids have cyclopentanoperhydrophenanthrin ring system. It is a fused ring system made up of three cyclohexane rings designated as A, B and C and a cyclopentane ring that is ring D. The six-membered rings are in a phenanthrin ring arrangement. Total number of 77 carbon atoms are present in cholesterol. On one hydroxyl group at third position, which is characteristic of all sterols. The OH group is beta oriented, projecting above the plane of ring. The double bond between carbon 5 and 6 is present. The 8th carbon atom chain, beta oriented, attached to 17th carbon chain, carbon is present. There are several centers of asymmetry, and therefore the possible numbers of isomers are many. One such isomer is produced when the double bond is between carbons 6 and 5 and 6 of cholesterol is reduced. Two possible isomers may be formed, example cholesterol and coprostenol, where the hydrogen atoms is beta oriented, projecting above the plane of the ring. In this classification, we can see the different cholesterol. First, cholesterol. Total number of carbon atoms is 27. Number of carbon atoms in side chain is 8. Its importance is that most important animal sterol. Then second is bile acids. Its total number of carbon atoms is 24. Number of carbon atoms in the side chain is 5, and its importance is it is a emulsifying agent. Then third is glucocorticoids and mineralocorticoids. Total number of carbon atoms is 21. Number of carbon atoms in side chain is 2, and its importance is that it influences metabolism as well as fluid and electrolyte balance. Number 4 is testosterone. Total number of carbon atoms is 19, and its importance is that it is a male sex hormone. Estrogen. Total number of carbon atom is 18, and its importance is it is a female sex hormone. Here ends our session. Kindly like, share and subscribe the channel. Thank you.